Okay, guys, so today we're going to be looking at water reabsorption in the kidney, and it's really key that you do follow this, and obviously we'll go over it again in year 13. So last lesson, just reminded me, we looked at the structure of the kidney, and we talked about the ultrafiltration occurring in the Bowman's capsule. We talked about the 3 levels of structure you need to know about the Bowman's capsule, which is the epithelial cell of the glomerulus, the basement membrane, and the endothelials podocytes of the Bowman's capsule. We then talked about the proximal convoluted tubule, where a lot of selective reabsorption occurs, and we said that from the kidney back into the bloodstream would be all of the glucose, 100% of the glucose, okay, and some of the sodium ions, some of the water starts moving through, some urea goes back out, okay, but all of the glucose is the key bit there. So just to remind you, we said that 45 centimeters cubed of fluid approximately every minute leaves the proximal convoluted tubule and enters what we're about to study called the Loop of Henle. That is still highly too much volume. You can't produce that per minute. Think about how much urine you produce a day. That is far too much water loss. So we're going to enter now something called the Loop of Henle, which is a U-shaped tube, and it's in the medulla of the kidney. And basically, it uses something to create it uses a mechanism to create a high salt concentration in the tissue fluid, which is the fluid around the loop of Henle, and that causes water reabsorption due to osmosis. Okay, so let's have a look at it and go through it. So basically, then you've got this structure here, which we're going to look at, and actually a bit further along into the collecting duct. And it starts in the cortex of the kidney. So if you remember, that's the outer bit of the kidney, as we talked about last lesson. Okay, and then it goes down into the medulla of the kidney, and it goes round and then back up through there. So now we've got to just introduce these 2 terms, which are really important. Okay, so the first part of the kidney, the bit I'm doing in green now, is called the descending limb. Okay, and then the second part of the kidney is called second part of the loop of Henle is called the ascending limb. Okay, and in order to make this work, we actually need to look at the ascending limb, the red bit first. Okay, so always think of an elevator, make sure you get those right around. You descending elevator, your ascending elevator. So we're going to look at the ascending limb first, and that is key to understand what happens in water reabsorption. If we have a look at the ascending limb, you need to know in the ascending limb, the walls are impermeable to water. So what that means is water cannot cross through the walls of the ascending limb. Okay? Cells, however, in the ascending limb, so the epithelial cells, actively transport sodium and chlorine ions out of the fluid, out of the nephron fluid, into in the tube into the tissue fluid. So we'll look at that in a diagram in a second. Okay, and this causes up to 4 times higher concentration than normal of ions in the tissue next to the descending limb. Okay, so really bad diagram here, but useful to show you. So what we're saying is this part, the red part, is impermeable to water, so no water can leave there. The water just literally cannot get out. The water just goes up and it keeps floating up, and nothing actually leaves. Okay, but ions can leave. So the sodium ions and the chlorine ions, which are the main 2, are actively transported, actively pumped into the tissue fluid, which is in this gap here. But it also, because I wanted to mention it now, happens this way as well. But we'll look at that later on. Okay. Now, what that is going to do is it is going to lower the water potential in this area of the tissue fluid here, because you're pumping all those sodium and chlorine ions out into that tissue fluid area. So as a result, water, as you know, will move from a higher water potential to a lower water potential. So water will move via osmosis from inside the descending limb out into the tissue fluid. Okay, so the descending limb is permeable to water as well as ions. Okay, and water is drawn out of the descending limb to the lower water potential, and some ions will move back into the tube as well. Okay, so the water moves out, which is what we're trying to do, conserve more water. Now, what that means, and I think this is a good time to show you, is the highest concentration is here, okay, in the loop of Henle. that is the point where you would have in the tissue fluid anyway, the lowest water potential or the highest concentration. And that is because at this point you've lost the water. Okay, so your water has moved out via osmosis, but we've got all the ions and all the sodium and chlorine ions. But obviously sodium and chlorine ions are going to actually start moving out by active transport, and that will then raise the water potential slightly as a result. So the water potential will increase, it will get higher or less negative as it goes up the ascending limb. So again, just different way I showed it. We actively transport sodium and chlorine out of the ascending limb, which is impermeable to water, but we have active mechanisms of carrier proteins to pump out that active transport of sodium ions into the tissue fluid. And there's passive at the bottom as well, should have said. That causes water to occur, to move out via osmosis, okay, and that causes a concentration highest at the bottom because you've got the lowest water and the most ions in it. And basically animals that live in the deserts and things like that will have a longer loop of Henle's, and the desert rat, I believe it is, has the longest one in proportion to the shape of its body, size of its body on Earth, but it has basically a third longer than human ones. So again, different ways of showing it. I think it's just really worth sort of showing you this diagram as well. So we've got, again, to summarize what you need to know. Loop of Henle starts in the cortex, goes down into medulla, and obviously ends again in the cortex. The ascending limb is impermeable to water. And the ascending limb, at the bottom of the ascending limb, sodium and chlorine come out via diffusion, which is shown in red, okay, because they'll start to come out there at first because there's a higher concentration. But then at the bottom, as you go up, they need to be actively transported. That causes the descending limb to allow water to leave via osmosis from a higher water potential to a low water potential into the tissue fluid, and that means you lose a lot of the water, and then that obviously goes back into the bloodstream. Okay, now what we're going to look at now in a moment is all of this also happens the other way. So the sodium ions also get transported, and the chlorine ions also get transported into tissue fluid here. So that also, once again, lowers the water potential here. Okay, and that means that once the water potential, because you've pumped all this stuff out, as you're up here, you might have less water, but you, once again, because you pumped all the sodium ions out and all the chlorine ions out, you actually have a relatively high water potential again. So whilst it's shown here just at the end over here, this then means more water can start moving out as we go through something called the proximal convoluted tubule, the distal convoluted tubule, and the collecting duct. As we continue along the loop, okay, as I just said, fluid becomes very concentrated. It starts the ascending limb, but because the ions leave, the fluid concentration starts to drop basically, and therefore it becomes gradually less concentrated. Okay, now you should be able to explain then what happens along these different stages. Okay, so if we have a look at this graph, this is a really complicated graph, but one that you do need to try and be able to explain. So what is happening in this graph then? Well, this shows you the different parts of the kidney nephron. So you've got the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. So obviously at the start, we've got the Bowman's capsule, which has done selective ultrafiltration here. Again, the concentration they've said here is concentration relative to the concentration in the glomerulus filtrate. So basically what they're saying is it's all at one. The glomerulus filtrate is all the things that come in the Bowman's capsule. So if we look at glucose, glucose is the easiest to explain what happens. So as you go through the proximal convoluted tubule, through the first bit where selective reabsorption happens, all of the glucose in a healthy person will be reabsorbed 100%. So that one I think is relatively straightforward. We don't need to hopefully explain that massively as a result. Okay, if we then have a look at what we got now, so I'm looking at the levels, sodium and potassium. Okay, let's have a look at this. So first thing then is the sodium levels do not change because there's no actual reabsorption of it during the proximal convoluted tubule. But in the loop of Henle, the sodium levels actually increase, or the relative concentration increases compared to the glomerulus filtrate, and then it drops. Now, why would that be the case? Well, this is relative concentration of sodium. So that is the case because as you go into the loop of Henle and you go down the descending limb, water is removed, and that means the concentration, as we said, becomes the highest at the bottom of the descending limb. But then the concentration of sodium ions starts to drop because the sodium ions are actively transported out and diffused at the start of it, but diffusion and actively transport out as you go down the the ascending limb of the loop of Henle. You can see the lowest point is here, which is when the change colours here, see what I'm saying? The lowest point is here, which is where at the top of the ascending limb, so that's where you stop pumping it out. Then, because water is continuing to be removed, okay, so all of this is just due to water removal, the concentration of sodium slowly increases because we're removing water in the last few stages. And the same with potassium. We don't talk about potassium at all. The only reason potassium concentrations increase again is because water is being removed, so therefore all the ions that are present are increasing in their concentration. Now, in terms of urea, okay, urea is going to slowly increase in concentration, and the reason urea is going to slowly increase in concentration is because we are removing other things other than urea. It's slow in the proximal convoluted tubule because some of the urea leaves as well, but as everything is removed, your urea concentrations are going to ever increase because we never remove it actively at any point during the process. The distal convoluted tubule and the collecting duct, then you need to just know that fluid then goes through that after it goes through the loop of Henle. It's very short, but it's got lots of microvilli, and it uses membrane pumps to pump more sodium out. Okay, and basically sodium ions are actively pumped again into the tissue fluid, and that means that water then will pass out as a result, and the potassium ions will also be actively pumped out. Sorry, potassium is actively pumped into the tubule from the bloodstream at that point.